So for viruses, it's a pretty short unit. Um, you have to remember that viruses aren't really determined living things. You could argue it either way for IB, but generally they're not. And that's because they don't really eat, they don't have excretion, and they're unable to survive without a host. So if you define living as has DNA, can reproduce, able to grow, then technically they are. But if you think about cell theory, then no, they aren't. So no virus can live freely, and they must be within another organism, meaning a host cell, and they are technically parasites. They have no nucleus, no cytoplasm, and no organelles and such. No membranes. They just have a nucleic acid core, which means they do have genetic material. They have either DNA or RNA, but never both. When they do have RNA, they're called retroviruses, such as with the case of HIV. They're covered by a protective protein coat called the capsid layer, and they reproduce by replicating through the host cell. And this is only within a host. Their sizes range from 10 to 400 nanometers, which is much smaller than bacteria, or single-celled organisms. But if a question asks you, they are bigger than the thickness of the cell membrane. I saw this in a paper one. They're all infectious, like no matter what. So, for example, there's the polio virus, rabies causes hydrophobia, where you're scared to swallow your own spit and drink water. Influenza and flu. Influenza is the virus. So, and then there's warts, herpes, or cold sores from the herpes simplex virus. HIV, human immunodeficient virus, causes acquired immunodeficiency syndrome. So AIDS is not the virus, HIV is, and Ebola. Here's the structure of a virus. So it looks kind of like a, I don't know, like a game thing, a game sprite, as this capsid coat surrounding the DNA or RNA, and it has this little spring coil for a tail sheath, and then this like table looking thing. And at the bottom, there's a spike, which is what it uses to insert DNA or RNA and make it through the cell membrane. These are the tail fibers. They're used to move around and attach onto the host. This one specifically is a T4 bacteriophage, and phage in this case means virus. So viruses are very specific. They're really picky. So they can only, so three types of viruses here. There are animal viruses, plant viruses, and bacterial viruses. And they can't interchange. So a bacterial virus will never be able to attack an animal virus. So those are called bacteriophages. And then there's the life cycle of viruses. There are two types of life cycles. There's the more fast-paced lytic cycle. Then there's the dormant lysogenic cycle. So the lytic cycle, lytic as in lysis, the death of host cells. So after the phage attacks, lytic refers to it rupturing the cell as it rapidly produces components within our cell and bursts our cell. And when it does that, the components will all attach onto nearby cells, causing a virulent cycle, meaning it's very fast spreading through your body, and it'll be fast spreading through individuals too. And then the lysogenic cycle, also known as the latent infection or dormant. A virus infects a cell but does not produce a lytic cycle right away. The virus remains latent or dormant. That means it remains inactive within the cell until some kind of stress or some kind of activation condition is met, and the cell actually suddenly displays signs of this cycle. So I believe there was a better handout that showed the full stages. But for the lytic cycle, there's infection where it attaches, so you can draw it attaching onto the surface. There's infection or growth where there is a hole open through the spike, and the contents, you can draw them injecting into the cell. The viral DNA will take over the host cell's metabolism and function. Maybe you can point it towards the nucleus. Replication, the host cell DNA is altered, the genome, so it, instead of producing itself or more organelles, it produces viral parts and proteins. And the host cell will fill with replicated viruses until it finally bursts, releasing up to 300 viruses that will then spread to other cells. And the rupture of host cells are seen as dark spots called plaques. And I got that question wrong. So the lytic cycle may take 30 to 45 minutes, and this is known as virulence. For the lysogenic cycle, in summary, it's quite similar to the lytic cycle, except they may be attached to the host DNA like an extra gene, and they will slowly produce it over time rather than immediately. So the combination of host cell DNA and viral DNA is called a prophage. As host cell divides, daughter cells also receive viral DNA. So it's like kind of a like an even bigger issue, because even the after mitosis, the further new cells will still have viruses in them, and it can be latent for up to even years until mutation, stress, etc. activate it. HIV, very important. HIV is a retrovirus, meaning it has RNA. It needs reverse transcriptase to be able to turn its own RNA into DNA for it to be able to add to the host genome. And that's it for viruses.